Good morning, students. Today I am going to talk about microscopy. How many people know about microscopy? Okay. Any idea? Yes. The object which increase the size of the tiny molecule, tiny particles. Right. Okay. Now come here. Microscopy. It is a machine or called instrument which magnify and resolve the object. Okay. Anybody have any idea about the magnification? Okay. Means degree of enlargement. Increase the size. Okay. So see here. Suppose this is a object. It is very small. Micro means beyond our eyes limits. We can't see through the naked eyes, unaided eyes, which we can't see through the naked unaided eyes. Means without any object, we can't see through these objects. Okay, but when we use this microscope, a specially designed device or my instruments, so it is short form microscope. So it's look like this. So it magnified. Okay, fine. What is resolve? Resolve means distinguish two closest point separate. Oh. Suppose here we can't see how many dot, what is the color, and what is the exact shape. But when we use this microscope, then they look like this, and they are separate, distinguished. So this is called resolving power of the microscope. Resolve means resolving power, magnify means magnification. Okay, now come. Any idea why this microscope is very important? Why this microscope is very important? Any idea? Okay, because this is the principal instrument of any biological laboratory, chemical laboratory, or any lab who work on the structural aspects or developmental biology or parasitology, medical science. You know test tube baby? Why test tube baby is nowadays very popular and easy to develop? Because this peculiar kind of microscope. So if we discuss about the use of this microscope or application of microscope, so what is the use? See, in medical, cell biology research, reproductive biology research, plant sciences, pharma, structural biology, huge plethora of n number of application, means A to Z alphabet. Okay. So any idea how we increase the resolving power? Okay, good. To reduce the wavelength of the light. If you reduce the wavelength of light, then the resolving power is increased. A good resolving power means better the microscope. Okay, fine. Anybody have any idea how to calculate resolving power? So I only draw a outline. I'm not drawing the whole microscope. So the The part which near to eye is called ocular lens. Ocular lens, so called eyepiece. Anybody have any idea what is the magnification power of the eyepiece? Okay, our eye is considered as a x. Human eye considered as a x. The eyepiece resolving power is either 10x or 20x. It depends on the type of the microscope. So there is another lens near to object, so called objective lens. So what is the magnification power of the objective lens? There are different type of objective lens. They start from 4x, 10x, 40x, 100x. Nowadays 200x is also available. Okay, but when we are using this 10x, 100x, then we need oil immersion lens. So my question is how one can calculate the magnification power. So very simple, just read the magnification power of the eyepiece. If it is 10x, and read the objective lens which is on the object. If it is 40x, simply multiply them. 10x multiply by 40x, it is 400x. So by this way one can calculate magnification power of the microscope. Okay, so tell me the highest magnification power of a light microscope. Okay, it is 1000x. Okay, so this is a fundamental things about microscopy. So we add some new things. Okay, so microscope basically divided into two category. One is called light microscope, light microscope, light microscope, and electron microscope. Electron microscope and light microscope, they are totally different microscope. Light microscope, here simple glass lens are used. The source of light is lamp or natural light, sunlight. With the help of mirror, we pass through the condenser, and it reach to the objective lens, and this pass to the till reach to the eyepiece. And in electron microscope, they have light is source is electron beam, and here we are not using glass lens. Here we are using electromagnetic lens, and the resolving power are much higher than this light microscope. Light microscope's magnification power, as I already discussed, 1000x. But here electron microscope, the resolving power is 50 million x. Nowadays the most powerful technique available on this earth in microscopy is cryo-electron microscope. Okay, so you have a question in your mind, how this sample prepare for the microscopy. So there are two. One method is manual method, you can just cut the slice, thin slice of your section, plant, animal, which is to according to your need and your requirement. So nowadays there is automated machines are designed to cut the section, so called microtome. Microtome. Microtome means this they cut this the special kind of protocol required to prepare the log, and then this log is cut by using this machine called microtome. Here you can adjust the thickness of your sample according to your choice and need. 5 microns meter thick sample is good. And another microtome is called ultra microtome. When sometime what happen, you want to visualize a peculiar kind of proteins or gene expression in your section, then or some special treatment you want to see or in special structure, so you need to cut in the by using ultra microtome. And ultra microtome is operated on 4-degree temperature. Okay, so once you cut the slice of your section, then you use this microscope according to your need, and you can got the good result. So nowadays these mitochondrial disease and chloroplast based disease or any pathogen, even virus, why the virus is easily predictable? Because of this electron microscope. When, oh, okay, fine. Anybody have any idea about the history of the electron microscope? Because these light microscopes are very popular, you know, because 16th century is called golden age of the biology. 16th and 17th century, because at that time Anthony von Leeuwenhoek and Robert Brown they discovered cell. Robert Brown in Robert Hooke, they use cork cell, and Robert Anthony von Leeuwenhoek, they use living cell, and they visualize the living cell by using this self-made light microscope. But the electron microscope discovery is very unique. It was invented by Knoll and Ruska, and they got Nobel Prize for this peculiar invention. No doubt. If there was Nobel Prize, then these two people, Robert Hooke and Anthony von Leeuwenhoek, they also received Nobel Prize. If they at that time there were Nobel Prize. So this is the peculiar history of this electron microscope. And when they invented electron microscope, then people are realized there is a virus, because virus range is nanometer. So in light microscope we can't see viruses. So even you can see the protein, DNA, by using electron microscope. So if you, there is a plethora, but there is a limitation in the application of this microscope. We never ever use any live material, live pathogenic material, first thing. Second thing, never ever use radioactively labeled material in electron microscope or in light microscope, because maybe it disturbs the instrument or it is the hazardous for the person who is operating this instrument, and here a special staining technique is also required. But electron microscope, there is a two different feature. One is called scanning electron microscope, other is called transmission electron microscope. So scanning electron microscope means when you want to visualize the 3D image, then you need a scanning electron microscope. If you want to visualize the 2D image, then use TEM, and the magnification power is TEM is much higher than scanning electron microscope. So now there is a small correction. Actually, I during my lecture I have used Robert Brown, not Robert Brown, it is Robert Hooke. Robert Hooke discovered first dead cell, means they used cork, and Robert Brown, the credit goes to Robert Brown for the discovery of the nucleus. So this is a small correction during my lecture. So this is a brief of or very superficial information about this microscopy, and in my next lecture I will discuss detail about the microscopy, each and every compartment. Thank you very much.